Hello, great students. Welcome to a wonderful week again. This week we'll be looking at the topic microorganisms in action. Our objectives this week are: 1, to define microorganisms; 2, to identify some microorganisms found in the environment, that is air and water; 2, describe various groups of microorganisms; and 3, to describe the beneficial and harmful effects of microorganisms on the human body. We should also describe vectors or carriers of microorganisms, and we should be able to measure or know how to measure the growth of microorganisms. And what are microorganisms? Microorganisms are living organisms that cannot be seen with the naked eye, but by the use of microscopes alone. We call them microbes or germs. It was first discovered by a scientist that we called Anthony van Leeuwenhoek in the year 1674. He used a simple microscope and he discovered rainwater that had been collected from pools was full of little organisms. That brings about a branch of biology that deals with the study of microorganisms, called microbiology. Microorganisms are ubiquitous in nature, which means that they exist everywhere, almost everywhere. We see them in water, we see them in air, we see them in the soil, under the fingernails, mouth cavity, inside ears, nose on the skin. You see them decompose organic matter. You see them even inside our food. They are found everywhere. In and out of the body of every living organism, you see microorganisms inhabiting. These are different areas that we can see microorganisms. Let's look at the groups of microorganisms. First one, we have the viruses. Viruses are non-cellular organisms without nucleus, cytoplasm, and cell membrane. They are invisible under the highest magnification of a light microscope. You can only see them if you use an electron microscope. The structure that we have here is the structure of a bacteriophage. A bacteriophage is a bacterial virus. It is made up of protein coats and the DNA. Now, the protein coats that we call the capsid and DNA that we call nucleic acid makes up this virus. Now, the protein coat is divided into head and tail. The head contains DNA. This is the DNA, while the tail region is being protected by a tail sheet. This is the tail sheet. The tail region ends with 6 tail fibers. It ends with 6 tail fibers. So some viruses also may have RNA instead of DNA, while some may have both RNA and DNA. These 6 fibers radiate from what we call the tail plate or the base plate. We have some examples of virus as influenza, herpes, mumps, poliomyelitis, smallpox, etc. Bacteriophage is an example of a virus. These are different types of viruses. We have bacteria as another example. Bacteria is a unicellular organism that multiply by binary fission. They have a cell wall. You see presence of cell wall, and they also have a capsule. The capsule plays 2 roles here. The capsule 1 protects the organism to avoid dehydration. 2, this capsule helps to provide a site for attachment of the organism to attach itself on the body of the host. These are the 2 functions that the capsule provides. We can see proteins, we can see the cytoplasm, we can see the mitochondria. So apart from that, we have strand of DNA that comes together to form a nucleoid. We have flagellum, this structure of a bacteria. After that, we have the classification of bacteria. One, classification based on shape. Bacteria, they are classified based on shape into three. We have the bacilli, we have the spiral, and we have the cocci. Now, based on this, we have bacilli. We have are the ones that are in their shape are in a rod-like shape. This rod-like shape, we have those that they have a long strand of rods, different rods shaped like bacteria that clumps together. Here you see the bacillus. Here you see the ones that are rod but they are flagellated. Here you see the flagellate rod. You see the spore former. Here you see the Clostridium botulinum. We have those that are spiral in shape. Here you see the vibrio cholerae, the cause of cholera. Now we have those that are spiral. As with the spiral, they always in form of a common-like shape. Why we have those that are in a spirochetes, where you see the Triponema. These are the ones that are in a spiral-like shape. We have those that are in sphere-like shape, where you see those that are in two conical joint, two sphere joined together. We say that they are diplococci. We have the streptococci, long chains. We have the tetra, 4 chains. Why we have the staphylococci? We have the sardina. So staphylococci, they call it, this is the one that is responsible for staphylococcus. So this one is responsible for streptococcus, which is pneumonia. Okay, so these are different classification of bacteria based on their shape. The second classification is based on their need for oxygen. Based on their use of oxygen for respiration, we classify them into three. The first one is aerobic bacteria, those that require oxygen for respiration. The second one, anaerobic bacteria, those that do not require oxygen for respiration. While the third one are the facultative bacteria, those that can live both under aerobic and anaerobic condition. That is, that these bacteria that we call facultative bacteria, they can survive whether in the presence of oxygen or in the absence of oxygen. Their respiration must occur. Another form of microorganism that we are going to be looking at is the fungi. Fungi, they are eukaryotic heterotroph. Some of them are parasitic, while some of them are saprophytic. So their body, they always have a vegetative filament called hyphae. So if you would, the combination of series of hyphae will give you what we call mycelium. While subdivision of hyphae will give you what we call rhizoid. Rhizoid is used for the attachment or absorption of nutrients from water. Apart from that, they reproduce by spores produced from the sporangium. A different way of reproduction in which that they can reproduce sexually and asexually. Asexually is by means of spores. We have examples like Mucor, Rhizopus, Penicillium, Aspergillus. These are different examples of fungi. Now let's look at the algae. The algae, they are multicellular organisms, while some also are unicellular. They are found mainly in water. Some are heterotroph, while others are autotroph. Like here we have Spiragyra as an autotroph, while we have Chlamydomonas as a heterotroph. Some are free-floating microscopic algae that we call them the phytoplankton that you always see in aquatic environments. You can see Protococcus. These are different examples of algae. The fifth one is protozoa, the fifth form of microorganisms. The protozoa, they are single-cell animals found in fresh water and moist soils. So they ingest organic food like bacteria, which is digested within their cell. The example of protozoa are Plasmodium, that causes malaria. We know that. We have Paramecium, we have Amoeba. Amoeba causes dysentery. While we have Trypanosoma, which causes sleeping sickness. These are different examples of protozoa. Culturing or growing microorganisms. How can we grow microorganisms? Or before we enter into how to grow microorganisms, the question should be, what is a culture? A culture refers to a population of microorganisms cultivated in an artificial growth medium. The artificial growth medium is a component on which microorganisms are cultured in the laboratory. We always call it culture medium. So if a culture medium consists of only one living species of microorganism, it is called a pure culture. Listen again, let me repeat. If a culture medium consists of only one living species of microorganism, it is called a pure culture or arsenic culture. Whereas if it contains two or more kinds of species growing together, it is called a mixed culture. The culture is always prepared with what we call agar. It provides the nutrient that the microorganism needs for it to grow. The steps that are involved. The steps that are involved in preparing a microorganisms or culturing microorganisms are: number one, preparation of culture medium. We dissolve a known weight of the medium into a distilled water or in a conical flask. You will sterilize the contents using pressure cooker, or you can even use autoclave. After sterilization, you will pour the medium into a sterilized petri dish and allow the medium to solidify. Now after that, you will start inoculation. Inoculation is the introduction of microorganisms into the prepared medium using what we call inoculating hook or wire. So at that time you are introducing the microorganism into the growth medium that you have prepared is what we call inoculation. Incubation, the time that you will start to put the culture under conditions that will favor the organisms to grow very well. You prepare a suitable temperature for it. Example, an incubator at 37 degrees for 48 hours will give microorganisms a stable temperature for them to grow well, for them to incubate. Now after incubation, you enter into identification of microorganisms. The colonies or the growth on the petri dish is being examined during this time for proper identification, because it's possible that different microorganisms have been cultured here. So a small portion of the colonies is smelled out. You bring them out on a slide and observe under a microscope so that you can be able to identify the microorganisms. Growth phases in microorganisms. Growth phases in microorganisms are into four. We have what we call the lag phase, we have what we call the log or exponential phase, and we have what we call the stationary phase, and we have the death or decline phase. Now I will explain this phase one after the other. You see, at the lag phase, what happens at the lag phase is that the organisms will start to adapt themselves to the growth condition. When you introduce them into a growth condition, they will start to adapt themselves into that environment. There will be an increase in size and mature, but they do not divide here and multiply. That's why you will see this organism here only one. There is increase in size and maturity, but they don't divide in size. Now after the lag phase, you will see the exponential phase. In the exponential phase or log phase, it's characterized by explosive self-doubling, fast division, in which there is growth at a maximum rate. The cell will start to divide, to double. After that time, you will enter into the stationary phase. In this phase, there is neither growth nor death. No growth, no death. As the population increases, there is competition against food and space, which will slow and stop the growth of the organism. Now here you will see that the organism, they are too crowded. What will happen? Competition will occur. Competition will occur. They will compete for food. They will compete for space. After that, death or decline phase will come in. The cells become incapable of growing. So the cells die off gradually. They start dying gradually. There will be a decline. There will be a decline where you see a long-term stationary. With this, you can see how different organisms, how in a culture medium, if a bacteria is in a culture medium, it will move from lag to exponential to stationary. From there, it will come down to death or decline phase. Ways of measuring the growth of microorganisms. We have the plate count. The plate count, in plate count, it involves cutting the number of colonies after each culture. While in measuring dry weight, in the measuring of dry weight, you can use a distilled water to harvest cultured microorganisms from the surface of the algae. Well, we have microscopic count. Here, a serial dilution is made and the number of microorganisms are counted using the microscope. We have the next one, cell density measurement. Cell density measurement, the density of an aqueous suspension of microorganisms can be measured using two substances or two instruments, either a calorimeter or a spectrophotometer. We have use of filter membrane. If one is using filter membrane to measure the growth of microorganisms, the microbes are cultured in a medium inside a membrane filter, and the number of colonies are later counted. It is always used for counting microbial loads in food, water and milk, or even soil. Now let's look at the beneficial effects of microorganisms. What are their beneficial effects? One, we have decomposition and ecological balance. We know that most of these microorganisms that they play a role in decomposition of organic matters. So they break down complex organic matters into simpler forms to add nutrient to the soil, and even something like nitrifying bacteria that breaks ammonia into nitrate. So these ones are the organisms that plays the role in decomposition and ecological balance. We have microorganisms as food. Fungi such as yeast, even mushroom, they are used as food. Even they are a very good source of protein. Some of them provide B vitamins. Some of them are low in fat and almost free of cholesterol. They are a very good source of food. We have in medicine, they are used in production of antibiotics. For example, some of them can be used as antitoxin and even anti-cholesterol. We know that some medicine, when we hear about ampicillin or penicillin, is being from a microorganism, penicillium. So they are very, very effective and they are very, very useful in medicine production of antibiotics. We have production of food. In production of food, something like Saccharomyces is always good for the bakers. It is known as the baker's yeast. They use it as a resin agent. Even for fermentation in bread making, they use microorganisms in order to achieve their work. Even some bacteria are used in production of food, such as cheese, soya sauce, vinegar, wine, even yogurt. Through fermentation, they can be able to use bacteria like lactobacillus or streptococcus. These bacteria, they play major roles in this area. Curing of tobacco. They are useful in the curing or ripening of tobacco leaves. Even in silage making, they also play major roles in preservation of pasture crop for future use. Here, what are the harmful effects of microorganisms? They will destroy our food and materials as well, because most of them, they spread about spoilage of food. Some fungi like Aspergillus, Saccharomyces, they bring about decay of our food. Other materials that are susceptible to deterioration by microorganisms are something like paper, textile, wood, rubber, even metals. They are being destroyed by microorganisms. The second one, which is cause diseases, just like as we know today, the COVID-19, the coronavirus is one of the microorganisms that is trembling today in the whole world. So it's a microorganism. It's causing a negative effect. So here we have list of different microorganisms. We have flavivirus, we have poliomycete, we have all of them. Now this will come as your assignment. I have listed all these microorganisms, the diseases they cause. Now your assignment will be to put the mode of transmission and their prevention mode. Mode of transmission and their prevention. I advise that you do it very well. Minimize copying from the internet. I always advise you guys, cite more of your textbook and notes than the internet. Thank you.